similar are these sequences? If I look at one sequence compared to the other, how can I tell how similar they are? And so this is a central piece of bioinformatics technology. I call it technology, but I mean, it is. It's a central piece of theory. And in bioinformatics, you're always taking one sequence and you are comparing it to another sequence. So that brings me then to part two, where we're going to be talking about, hang on a second, we'll get to the slides. But if you go to the, when we're looking at, like, for instance, different types of organisms, we are always comparing one type of organism with another. And so in today's class, I have some examples of actually making some physical observations, and then we're going to take that into the kind of, comparing genetically. We're going to be doing some discussing genetic comparisons. But basically, though, I think that the argument that I'm going to make will be hopefully very compelling. That you'll see that that you really are spending a lot of time in bioinformatics just comparing things. You basically learn something, and then you use that knowledge to learn more stuff by comparing how much of that knowledge you have to how much new knowledge there is. And then the new knowledge, the stuff that you discover, is maybe your new research area. So anyway, let me just start with this. I'd like to start with descent with modification. And what we're really talking about is, the origin of species, one of, I guess I would say, one of science's best books, which is Charles Darwin's book, where he went to the Galapagos Islands, and he was looking around at some of the animals on these islands, which really were evolving kind of separately from the other types of islands that he'd been to. I mean, he was from England, and so they're actually very different. The evolutions that were there were actually kind of different from the evolutions in England, but he still put everything together. And what he put together was that when he saw a certain type of finch that lived on one of the Galapagos Islands, he found that that same type of finch existed on another island, but with modifications. And so he began to go through this whole theory in his head, thinking, how is it that I'm getting these two different types of birds that live in that look very similar, but they're living in different environments, and they are kind of becoming a different type of species. They're becoming a different type of bird. And so he called this his theory of evolution. And if you ever have if you ever have like a good couple of weeks here, I do recommend that you pick up his book, The Origin of Species, and give it a read, though, because it is probably one of the finest works of science, I argue, that ever was. Next to, I would say, probably Isaac Newton's Principia, which is his calculus. But anyway, these are some of the ideas that he came up with, and that is that that basically you're getting a gene which is passed on from species to species, or from parent to offspring, and if that offspring is somehow, if it helps the offspring's survival rate to have that modification, then it stays. However, if it does not help the offspring, if the offspring dies, or if the main organism dies, if the gene in some case, or the adaptation kills the organism, then it does not go on. It just stops. It's basically it's a what they call an environmental pressure that forces change to happen. But if that change doesn't do the things that you want it to do, then it doesn't persist. So we're really talking about changes that are being passed from genome to genome, from parent to offspring. And so when you're getting the genes from your I guess your parents, or when the offspring gets the genes from its parents, the DNA is basically, I mean, it's faithfully copied. When I say faithfully copied, I mean that it's basically copied. It's almost the same kind of, the genes you get from the parents are more or less the same, but you do get these mutations. Now, we've spent some time talking about mutations, and the thing is, though, that we don't get exactly 100% accurate replication when we get the DNA from our parents. Well, for one thing, you get DNA from your father, you get DNA from your mother, and those two different types of DNAs come together and they intermingle, and then that becomes your genetic. So in some very real sense, you are entirely unique. There is nobody who has the same genetics as you. I mean, you're all unique. So the question that I'm asking here, though, is that what would happen if you did get 100% accurate replication? That means that you did get exactly the same genes that your parents had, with no modification, no mutations, no nothing. Well, if that were to happen, then you wouldn't be able to succumb to, you wouldn't be able to resist environmental pressures to change. You wouldn't be able to change with the environment. So, for instance, if you were one of the Galapagos finches and you needed special a special beak to be able to get into the types of seeds that you need that come from a special fruit that's on the other side of the island. Supposing if you didn't work, if you had to develop maybe better wings to fly there, and then a better beak to be able to actually break down the fruits that have these seeds that you want. If you weren't able to make those modifications, then you wouldn't be able to feed, and then your species would die. And so it's a high. So is high similarity really? When I say high similarity, is it really desirable to have exactly the same genes as your parents? No, it is not. And so that is a good thing because you want to be able to deal with environmental pressures as they come. For instance, I mean, look around. All of us here, we all use oxygen. We all use oxygen. But there was a time when life didn't do very well with massive amounts of oxygen that we have in the planet. We just didn't. In prehistoric times, there wasn't much oxygen, and so if you were exposed to oxygen, you had oxidation, which was like rust, and so that was a dangerous thing. And so we have evolved as biology to be able to withstand oxygen in our environment. In fact, we thrive on this. We use oxygen. Just matter of fact, it's cool. This morning I was like just taking in so much oxygen as I was going for my walk. It's like you just need that oxygen. So we call this descent with modifications. As you move forward, you're basically your genetics are changing. One of the most unusual examples, but I mean, when you look at it, it makes sense. But for some reason, this eluded many people before Darwin's time, and that is that. Did you know that whales are actually types of cows? They're actually related to cows. They're mammals. This is a cow that swims around in the ocean. Whales. In fact, whale babies are called calves, and they do have hair, which is interesting. I mean, what fish can you think of that has hair? And so, if you look closely at some of the bone structure that exists in whales, you'll actually notice that at the bottom here, a long, long time ago, there's this animal that looks kind of like maybe a muskrat or something, but it's obviously it's a mammal. It's obviously a mammal. It has the same kind of 4 feet on the ground. It has a tail. It has a vertebrate, as we know mammals to have. This mammal suddenly realized, or not suddenly, but over the course of evolutionary time, this mammal realized that maybe there's more food to be had in the ocean. Maybe there's more resources, or maybe better survival in the ocean. So it began spending more and more of its time in the ocean, and then now today it's called the whale. And so it's the modern toothed whale, and it used to be living on land. But one of the interesting things is if you look at some of the bones that these animals have, it's like you can see that there is this descent by modification that actually does happen. This bone that you see here, which is the orange one, is in human, cat, whale, and even bat, which are very different types of biology. I mean, they're very different organisms in terms of, well, seemingly physiology and diet and how they live. But they have the same bone that exists here. This is I guess it's your arm bone. And this bone in your arm exists in a cat's leg. It also exists in a whale's fin, which also exists in a bat's wing. And so this bone was actually left over from the time when we were all the same species, but then we kind of forked out, I guess, and we went on our own journeys to become different species. So that suggests that human, bat, cat, whale organisms were actually one organism at one time. But most incredibly, though, one of the most incredible things that I keep looking at this thing, just scratching my head thinking, what an amazing thing. And that is that whales have a pelvis bone, which is, like you're sitting on it right now. If you're sitting down on your seat, you're sitting on your pelvis, which is this bone down here. And we have these things. But whales also have these things, and they don't seem to use them at all. In fact, these are just free-hanging bones, if you will, that are just kind of like embedded in the blubber somewhere. That doesn't sound very, very friendly to our whale friends here, but it's true. They're just bones that are left over from the time going back when this whale was actually this hypothetical, muskrat-like creature. Anyways, I think it's quite incredible. And so we talk about like this descent with modification in terms of like dogs. In fact, all dogs or that we see today are in some way related to the gray wolf. I mean, they're all coming back from, they all descended from the wolf, and it makes sense. I mean, a lot of dogs are very similar. They have ears that are big, droopy, cute ears, and they have a cute little fuzzy faces, and they're just fuzzy little bodies. It's like they're just they're all the same kind of cuteness that must have come from some common place, and that comes from these gray wolves. And so we call these dogs like subspecies of these gray wolves. There's one organism which branched out into other types of dogs, and then those dogs became the organisms that they are. Now, the thing is, though, that these dogs were probably, the environmental pressures that these dogs received probably came in the form of humans. I know that beagles, for instance, back in ancient, I mean, I have a beagle, which is, they're remarkable companions. He's a really wonderful guy, my beagle. But his ancestry actually comes from, I think, ancient Mesopotamia, where people were cultivating different types of dogs to go on hunts and to smell things out and everything. And beagles became a species that was in that cultivation. Other dogs have similar stories. You know, they're all bred for different types of things, and so that breeding made them into the animals that they are. And so I think that we can safely say, though, that you have this kind of a tree of life where there's one gray wolf, and then all these other dogs kind of came from that. Now, in terms of the sequences, that you can tell that these dogs were all related to each other, because if you go back and look at the gray wolf, and the gray wolf still exists. By the way, let me just say something here. The gray wolf still exists. A lot of people would think, you know, well, if all these dogs originated from the gray wolf, then why is it the gray wolf still exists today? I mean, why didn't the gray wolf just disappear and become a beagle or something? Well, that's because the gray wolf was still able to make ends meet biologically in its environment. I mean, it was still able to survive. And so part of the species of gray wolves separated, and the rest of the gray wolves remained gray wolves. Part of that species, kind of, you know, half of the team, if you will, or the half of the pack of dogs, if you will, from evolutionary time, separated and became the other dogs. But you still have these gray wolves that were still able to survive. And because they were able to survive, then they were able to pass on their genes. Other animals that haven't passed on their genes are like the dodo, for instance. You know, Gone the Way of the Dodo, this bird from 1920, I think it is, or 1918 or something. Since photography was invented, there's actually a picture of the last great dodo, and this bird died. The last one died because of its environment was impacted, and it wasn't able to adapt fast enough. And it was probably a human thing, of course, but it wasn't able to adapt fast enough, and so therefore it wasn't able to live on and have offspring to kind of keep its genome going. But anyway, if you look at getting back to the dogs, though, the genes, though, all throughout all this time here, you know, the separation of you know, maybe the mastiff and the beagle, they're different because over time their breeding has given them substitutions or different types of mutations in their genes. So the mutations have actually changed who they are as animals just by creating new genetic code, basically randomly. The thing is, though, that where these adaptations helped the animals to become a better, faster species, like if you were able to run faster or smell better or find your food in a more efficient way, then that modification, if that's what that modification did to you, if it gives you some kind of, you know, competitive advantage, then that modification would stick around. But if, for instance, you had a modification in your genes, which could equally happen, I mean, you could get a substitution or an insertion or a deletion as a dog that would somehow prevent you from smelling anything. Well, imagine how far would that get you in finding food, and so that would not be an advantageous type of you know, enhancement to get. So you probably that wouldn't help you to survive any better. And so the survival that we see today is really it's what they would call, like, I guess, organic genetic engineering, where the stuff that you see in people's bodies or that animals have in their bodies, the genetics that organisms have, is actually refined over millions and millions and millions of years of testing, if you think. So getting back to this statement here about parents and children, it says here, you are unique and slightly genetically different. And you want to be slightly genetically different because you want to have the traits that your father brings to you, and you want the traits that your mother brings to you. And hopefully those traits will provide you with a better competitive advantage that you'll be able to use in this time so that you can live long enough to give these traits on to your offspring, you know, and then they can use them in their time to continue the circle of life. So anyway, so we're talking about, for instance, this genetically different stuff. But the thing is, though, that we're also going to talk about retroviruses. This includes influenza, HIV, even coronavirus. But these are also unique and slightly genetically different from their ancestors. And so while this difference in genetics was important in dogs and people and animals and plants and things, genetic modification here is also important for these viruses because that's also how they survive. So before we get into these genetic abnormalities that we'd use in these viruses, let's spend some time actually talking about what is influenza. Influenza is basically it's very, very similar to what we have in COVID. It's not COVID, but it's similar in terms of how it moves around, how it functions biologically, how it's spread, and some of the symptoms that it brings. So, for instance, you get it's like they call this a very, very strong flu. I mean, people have called coronavirus a very strong flu, but it is so much more and so much more lethal. Whereas influenza is still lethal, but perhaps it isn't as dangerous. I mean, I'm going on a limb here, but perhaps it's not as dangerous as the coronavirus. But still, it's flu-like symptoms: fever, headache, tiredness. You get a dry cough, sore throat, nasal congestion, and you get aches. And it basically targets like your respiratory system. So you have all this coughing that you're doing here. Your lungs are infected. You can get pneumonia where your lungs will fill up with fluid. It's really something you don't want to have. And so this brings us to the next question. Well, what brought on this thing? What is influenza from? Well, influenza is what they call a retrovirus. Now, I had to put this picture out here, this retrovirus, kind of a disco star, but it's really what a retrovirus means is that the virus's DNA, the virus doesn't have DNA really, it has RNA. And so when it infects you, it infects its RNA into you, and that RNA then becomes, it's then translated or transcribed rather back into DNA retroactively, and then it becomes this and that's how it kind of meddles with your DNA by inserting DNA that is created from its RNA. But one of the things about working with retroviruses, as opposed to viruses that actually have DNA, is that, as we know, when you're copying DNA during the transcription stage of things, your DNA, well, actually anyone's DNA in the cell, DNA has all these kind of proofread, or it's proofread. It's got error checking. If there's some kind of an error that exists in that DNA, chances are, many cases, some biological machinery is going to come along and fix that error. And so you have this proofreading ability in DNA. That is why, for instance, that while we are genetically similar to our parents, and we still are slightly different from them, because some of these errors, and I hate calling them errors because sometimes they're positive things, but some of those errors crept through, and those modifications have made us unique into what we are. So you have many of the cases of DNA, the errors are being corrected. But that's not the case with RNA. And so in fact, with RNA, what you get is this RNA code that's basically is copied from one thing to another in these viruses, but there's no error checking. It's just copied willy-nilly. And if you think about the reason how this happens, that is that RNA is one sequence, whereas DNA is two sequences. And so when you're copying DNA, if you believe, and if you're a piece of biological machinery here, I mean, there's a lot of things that are going on here, but if you're a piece of biological machinery that's undertaking transcription, where you're basically, I guess or actually not transcription, but it's like meiosis, I think it is, one of the cell division things, when DNA is being copied, during that copying stage, you have two different sequences. And so if a base has been knocked out on your helix, if one of the bases on one side of the helix has been knocked out, then you can always refer to the other side of the helix, to figure out what that missing base was. That's before the pieces have torn apart. I guess. I mean, there's other things that happen after that. But while the DNA is in a helix, if one side is somehow, if one of the bases is removed, then the biological machinery can simply just refer to the other side of the DNA. Now I'm glossing over a lot of theory here, a lot of discussion, but you have this reference point. You have two places you have two addresses for these bases, on one side and another side. And if one of those bases is removed, you check the address at the other side to see what it was, and that will inform the biological machinery about what was missing. But with RNA, it's one sequence. And so if something is missing, it's gone. There's no way you can go back and check because there's no reference point. You can't go and check another piece of RNA. There's nothing on the other, there's no helix, there's nothing. It's just half the ladder. And so that is in itself a problem when it comes to correcting mistakes that may happen. So you get mutations that show up seemingly all the time. You get many of these errors. So basically, influenza lacks the ability to repair errors because, as an RNA virus, it lacks the self-correcting mechanisms to check that the virus or the virus's RNA has been copied correctly. And so they call this, what is it called? Stability, genetically stable. They're saying that influenza is not genetically stable because it changes so quickly. It's not its fault. It's just that by the mechanism of using RNA as its main system of genetics, it just changes its DNA or its genetic material seemingly all the time. And that makes a problem because when you have these alterations happening, the alterations could affect seemingly anything. And in viruses, because they replicate so quickly, it's during that replication time that these mutations would happen. And if it happens to be that those mutations create a more advanced, more sophisticated virus, which is able to infect more organisms, then that helps the organism. And so that trait will be carried on and passed on into the next generation. However, if some kind of mutation happened where the virus somehow miraculously died, it didn't help it at all, and that could equally happen too, then that particular virus that had that trait dies. But the other ones that survived move on. In fact, in computer science, there's a theory called genetic algorithms, and that basically is the same idea where you have a whole bunch of solutions and you're just testing all of them. They're all basically random solutions for some problem, and the problems that or the solutions that work miraculously enough, just randomly working for the problem at hand, those ones stick around, and then you just keep on hashing out those ones, and then the solutions that did not work are let go. And so after a while, you're kind of sorting through, after a couple of iterations of the system, of running this kind of system here, you're kind of sorting through only solutions that work with the problem, and then you can start deciding, well, which solution do I want to use. Same idea with these viruses. After a while, the ones that only the strong will survive, and the stronger, and the ones and as they're surviving, they get stronger and stronger and stronger. So this is what they look like here. You have this H1N1 influenza virus, very similar to the coronavirus. These pegs that are on the outside, those are called the spikes, the protein spikes, and those are the things that actually stick onto the cell, and they're kind of like I guess keys that unlock a part of the cell. And once they unlock the part of the cell, then the cell kind of just lets in this virus. And by letting in this virus, the green stuff in the middle, this is your RNA genome. This is its genetic content. It's the RNP, or the ribonucleoprotein, which contains the RNA itself. This is also injected into the cell, and then once it's inside the cell, the RNA is converted or translated, transcribed back into DNA, and then that DNA starts making other proteins. I mean, basically, the proteins that it creates are all viral proteins from that point on. And those proteins then insert this DNA into the cell's DNA system, and then the cell starts producing more viruses. It's like your virus hijacks the cell. It's really cool to watch animation, videos of these things, but also it's very scary at the same time because you think, wait, my body is being held for ransom by some kind of a virus. It's actually very scary. Believe it or not, in 2009, there was an infectious disease outbreak, the H1N1 influenza pandemic. We didn't hear much about it because we were actually able, as our World Health Organization and some other, the CDC and some other big organizations, were able to come together quickly and to prevent this from becoming a bigger pandemic. But it was a pandemic. It was the H1N1 influenza pandemic. People were told at that time to wash hands, wear masks. If you had a compromised immune system, you were warned by your doctors, given certain vitamins and minerals and things. But also there were vaccines that came out. And that brings me to this. So, how did we stifle that pandemic? Why was it not worse? Well, that vaccine and the vaccine that we use today is a very similar type of idea, and that is that when you have your, let's go back to this for a second here. See on this virus molecule, you have all this stuff which is like all these spike proteins which are floating around on the outside. Those spikes then stick to a cell. They're like little suction cups that stick to the receptors on cells. And then once the cell has one of these things stuck to it, it doesn't. I mean, it thinks, oh, you must be a friend. I'll let you in. It's like that kind of thing. And so it opens up, and then this virus comes in. So what vaccines are, if you can see right here, they are basically producing what's called antibodies, which are your immune system's natural response to fighting off infection. These antibodies are produced in mass in your body, and then they stick, if you will, to these spike proteins which exist on the outside of these viruses. And so what does that do? Well, imagine, for instance, in this case here, this antibody is sticking to this purple thing, or this, I guess, rose-colored thing. That's the spike protein that would stick to your cell. But the thing is, though, that if there's nothing blocking it, then it can easily attach to the cell. However, if there's an antibody stuck in this receptor, right here, then it's like it can't stick to anything else because something's in the way. And so really what it's doing is, like, imagine that you're sitting down at a buffet or whatever it is, or you're at a wedding and you're sitting down at this, you just got your plate and you're sitting down and there's this empty chair that's next to you, and you're kind of hoping that somebody who's eyeballing you from across the room doesn't come over and sit down next to you. Well, because you have that chair that's vacant, that person could easily come over and sit down next to you, and maybe you don't want that. And so what you do is you get rid of that chair, and so there's no way that anyone can come and sit down next to you because it's physically impossible. There is no chair. So then you're able to munch through your buffet from the wedding in peace. It's the same idea. So these antibodies actually kind of clog the pores, if you will, or they clog the spike proteins of these viruses, so that the viruses cannot actually latch on to the cell. And the reason why that's or how that helps anything is because if the cell can't latch, or the virus can't latch on to the cell, then the virus cannot introduce its RNA that starts creating more DNA, that starts creating more viruses, which starts creating more infection. So you're basically preventing that virus from being able to get into anyone's cell, and so then your natural immune system is able to just destroy this foreign protein basically in your body. Yes, sir. So when they make the vaccines, they cripple there is some RNA in there, but it's just the RNA for the spike proteins. You can't actually. So you're talking about the coronavirus, I guess. So the coronavirus, actually, when they make the vaccine. So the question is, when you're making a vaccine, are you taking out like all of the RNA, or what exactly is in a vaccine? So in a vaccine, you have the RNA that creates the spike protein, because that after all is the point of attack in your immune system. You're actually going after these spikes on the virus. You're not going after the virus DNA, or anything. You're not trying to lacerate the virus in some way, but you're going after the spikes and preventing those spikes from joining onto a cell. And so when you get a vaccine, you're actually getting a piece of the RNA that would create that spike protein, such that your body can play and figure out how to kill or how to bond to these spike proteins, because after all, they are foreign proteins in your body, and so your immune system response would be activated. But the thing is, though, that while your body is still learning, while you're making the antibodies to combat this virus, there is no virus. It's like you're not going to get infected. So in other words, you're just target practicing. Your immune system is target practicing with these spike proteins, and that's really all that there is. And so if those spike proteins do happen to stick to cells when they first are initiated, as they will when they're first introduced into the body, the spike proteins will stick to cells. But that's it. Nothing more can come of it. There's no virus that's coming after that spike protein. So in other words, you're just creating the keys, and you're trying out the different, to see whether those keys fit in these different locks. But you can't actually turn the key and open the door. It's like there's nothing to come through the door. There's nothing. The rest of the virus isn't there. It's actually very exciting, and actually, if you think about it, vaccines. There's no way that we can handle vaccines in this class. That's actually a whole That's like 2 semesters worth of biochemistry right there, vaccine development. Um, there are classes out there that exist, and there's a bunch of things online that you can read about. I've put some videos in here, and we'll talk about them momentarily, which give you an idea about how vaccines work. But I do a good question though, very good question. Okay, so moving on to this. So getting back to the virus evolution here. So what about this this descent with modification stuff? Well, if you think about it, these viruses actually they change themselves all the time. Viruses have a well. I'll just read this over here. The mutation rate for viruses is much lower. That means you have, or sorry, you have. It's much more. Viruses will mutate more frequently than they will in humans. So I guess you call it like a higher mutation rate than humans, which is a lower mutation rate. Humans have DNA, and DNA has error checking, and so therefore we're not expecting to find as many. I mean, they happen, but we're not expecting to find as many mutations in typical human DNA copying as, for instance, when viruses go from, if they're retroviruses where they're going from RNA to DNA, that part makes a lot of errors, which none of them are corrected. And then once it's DNA, it's DNA. But otherwise, viruses actually that themselves also use DNA. They also create. I mean, they also have error checking, but they still mutate. And viruses will mutate because that's how they survive. If they don't mutate, in other words, if they don't allow themselves, if their error checking is so good that they're always a direct copy of their ancestors, then they will never survive the pressures of environmental or the environmental pressures that would cause them to, or they would ask them to, or force them to move into something more advanced. One thing I will say though that is interesting here is only 1.5% of the human genome is in coding regions. That's not very much. That means 1.5% of your DNA is actually creating protein. I mean, there's a lot of protein that you have that you're creating in your body, but there's equally, I mean, there's even much more DNA that you have in your body. So you're thinking, what is all this extra DNA doing? And that's a lot of extra stuff. And so if you think about how it is that mutations happen all the time, I mean, you just get them. You can't always stop it. I mean, you have error checking code or error checking ability to prevent errors from happening when you're copying DNA. But because you have such a little amount of DNA actually being used for coding regions, that means that those mutations would have to, first of all, be unchecked and also land in the right places of your genome such that you create such that some modification happens. Now, it does happen, as we know from our discussion in cystic fibrosis, it does happen. You do get errors. But for viruses, though, it happens like all the time, and they have much less vacant space. In other words, the entire genome is all in there. Is all genes. So that's what I say. When I say vacant space here, it's like they don't have like junk DNA. It's all genes, and so anytime they get a mutation, seemingly anywhere, it's a mutation. It's a change to their protein development in some way. Anyhow, so yeah, so DNA comes from viruses, and you have DNA coming from, or RNA coming from viruses, and so I think that no matter what the thing is, you still get all these mutations that occur because the DNA that you get in viruses, there's much less DNA, I guess, much less vacant DNA. It's all stuff that does something. It's all DNA code that does something. And so any virus mutations that happen there, if they help the virus survives and moves on and infects more cells. If that mutation does not help the virus to survive, then the virus dies and some other virus will probably get a mutation that'll help it. So then that will kind of start building on. And so the thing is, though, that viruses mutate. They still mutate. But RNA mutations are probably one of the worst kinds of mutations because they mutate so often. RNA mutates so often during the virus's life cycle that it actually creates an entirely different virus after some time, it seems. It seems that in fact, I have a slide here that's saying that with a tree, for instance, of all the coronaviruses that have been found, you'll find that they have so many different variants of the coronavirus. And that's all because they've just been changing. So another thing I should say though is like you're probably wondering like, well, where's all this change happening in RNA and DNA? Well, DNA, remember, is the cell's most precious possession. It's the most important thing that it owns, and so that means that the most important things that anyone would own would be placed into a safe. And in the cell's case, the most important thing that it owns, its DNA, is placed into the nucleus, which is like this special, access or high clearance place where nothing can go except for special, biological machinery that the cell authorizes, because that's where the DNA is. And if anything happens to that DNA, then the cell is dead. It's simple as that. If that DNA gets destroyed or attacked or cut up or whatever, if something bad happens to it, then the cell will die. However, RNA is outside of that nucleus. RNA is created inside the nucleus, copied and sent outside, and that's where protein begins to do something, or that's when RNA begins to make protein. The protein production never happens inside the nucleus. It happens outside the nucleus. DNA is copied inside the nucleus. That DNA, that copy of that DNA, or that RNA, I guess, after transcription, that RNA is then sent outside of the nucleus, and that's where the protein production happens. If you think about it, the cell is a very dangerous place where outside of the nucleus, it's just turbulence and things are bumping into each other. And so you can, so it's easier to leave your DNA safe inside the nucleus, which is relatively, it's compact and it's a safe environment. And then you can just send out your RNA that you copied from that DNA. And that's where the protein production happens. It's a wild and crazy place in the cytoplasm. But DNA viruses are stable while these RNA viruses are unstable, because your RNA just mutates all the time, whereas DNA has these built-in checks and error checking, but also it happens in a very safe environment, the nucleus, where you're not likely to find much bumping around. So this is the most unusual thing, though. But for DNA viruses, this viral code is injected into the host DNA, and so that means that some part of the virus is able to actually get inside the nucleus and start meddling with the host's DNA, which is actually kind of, it's like a major security violation. It's like you're suddenly getting to this vault and messing around with the code in in the DNA. It's like it's a major affront to the cell. The DNA viruses skip the DNA and they just start duplicating themselves. And once they have duplicated themselves, then DNA is made from that RNA to create proteins that do other things. There's actually a video that I have someplace in these slides. I'll point it out when we get there to talk about, or that gives you a really good animation about how this all comes together. But the thing is that these retroviruses, when they infect, they don't kill the cell initially, because if they did that, then they would not get the production assistance that they need to create their proteins. They basically take the cell hostage, and then the cell begins to produce viral proteins and all these other things that they need to survive. And so there you have it. And they're basically they're hijacking the cell. And so if you hijack the cell, if you kill it, the first thing you do when you get there, or if you absorb it or you digest it, then you can't use the qualities that the cell has to produce your own proteins. But they call this reverse transcription, where you're going from your RNA back into DNA. That doesn't usually happen in regular non-viral. I won't say that happens, but typically though you go from DNA to RNA. That's the direction. DNA has the genetic code. RNA is a copy of that genetic code. RNA goes on to make the protein. But here you're kind of going backwards, where you're saying, okay, well, here we have RNA. I'm going to go back and get the DNA, and then from that DNA, I'm going to start doing some other things and creating other RNA from there that's going to mass produce my proteins. But still, it's kind of a fluke of nature how this whole thing happens. But when you do that, again, I keep saying this, you have a high mutation rate of your genetic material coming from your RNA, because RNA has no checks, or it has few checks. And so these mutations, though, you're probably wondering, why am I spending so much time talking about this? It's because when you create all these mutations, you're actually creating different sequences that you can compare. And so that means that when you're comparing sequences using like the Smith-Waterman or the Needleman-Wunsch algorithms, global and local alignment, you're able to see how different these two organisms are and see whether could there have been a time when they were the same organism. This is actually a very interesting but sad type of table here. When I say sad, it represents loss of life. And so we must respect this data. But these are patients A, B, C through F who were all infected with HIV when they went in for a regular routine visit to their Florida dentist. I believe this happened like back in I want to say the 80s. But this is a time when AIDS, where HIV as a virus was really not understood very well, and in some medical practices, they didn't realize that AIDS can be spread in blood. And so there were patients who went to the dentist's office and they had their teeth checked and the instruments were not properly cleaned, and so they developed HIV. And so we see on the right-hand side here the average differences in DNA sequences. We're looking at the DNA which comes from patients, or DNA for this particular HIV, but in the DNA, not the whole genome, but a specific area. But we're looking at those areas which would be telling of HIV. In comparison, we're comparing those sequences across the patients who have HIV, and we're finding that patients A through D. Actually, if you look at, for instance, the dentist is 11.0, 10.9, which is the patient A, patient B. These are scores, just think of them as scores right now, of similarity, or those numbers, if those numbers are the same, those scores are the same, which suggests that those are actually very similar sequences. And so you're looking at those numbers there, which are very similar scores, and so you're saying that this is actually suggesting that the sequences from all of these patients are very similar. And that also suggests that they perhaps have DNA which may have come from the same types of origins. In other words, they all got AIDS from the same place because their DNA has similarities which are indicative of HIV. And so if you check somebody who does not have AIDS or whatever it may be, I think patient F, HIV from dentist, you'll see that the scores for patient A through, I'll say through G here, 3.4, 4.4, 3. They're very similar numbers. Obviously, there's a bunch of other science going on here, but they're very similar numbers. But patient F, though, I think I think they may have HIV, but it's a different strand of HIV than the strand that the other ones got from the dentist. And so really what this is telling us is that you can look at DNA and determine, for instance, where a virus perhaps came from. And here, these patients, they found that they actually got their virus from this Florida dental practice, just because it has similar DNA. Here are some interesting videos that I encourage you to watch. I'm not going to watch them in class, but I do encourage you to watch these things, though. It's talking about how viruses, like the lifestyle of the coronavirus, the life cycle. It's actually quite interesting. It's quite creepy if you think about all the things that are going wrong, or right for the protein, or right for the virus, but wrong for us, that allows us to become infected by these viruses. But it's all random stuff. I mean, it was all just put together over millions of years from environmental pressures, and if something works, then that thing made the protein stronger, and that protein may have helped the virus, so the virus became stronger, and so therefore it stuck around. So what I'm saying is, all of these adaptations, if they helped, they stuck around. So going back to this alignment here, in fact, I have to get this thing done kind of quickly here, we're running out of time. But what we're doing is we're looking, and we've seen this slide before, but we're looking at different viruses, and we're running a similarity test, which is basically this pairwise alignment. And we're thinking, is there any way to determine whether or not these sequences are similar? And there is, and you use this alignment. You're using the Smith-Waterman or the Needleman-Wundt, but you're determining whether, for instance, these two viruses that I have, are they perhaps the same virus? Or, for instance, is one virus just a slight mutation, a slight mutationed variant of the other? And if it is a slight variant of the other with mutations, how many are there? How close is the match? And so we use this pairwise alignment. This is what we spent all last week doing. You'd use the same technology on larger sequences to determine how related that they are. I just have these slides up here, coronaviruses, just to give you an idea. But when you're talking about coronaviruses here, maybe, in fact, let me just go down to the end for a second here. This table here is actually kind of a mysterious looking table. But actually what it is it's saying, looks like my text was cut off here. But what it is like this is a tree of the variants of coronavirus. And so on the far left-hand side, we see like 2019, December of 3. We would call that the roots of this tree. But the thing is though, that this is basically where they began checking or testing or collecting data about the coronavirus. This doesn't mean that the coronavirus didn't exist before this time. It existed, but perhaps it wasn't medically relevant to collect data. But then after December 2019, they started keeping information about the coronavirus. That's why they call it COVID-19. The 19 is the year that it was discovered. That doesn't mean that there was like 1 through 18 versions of this before, which is a common misconception. It's the year. So these are all these variants. And so what I've been putting out before you today is like kind of where do these variants come from? These variants came from the fact that the coronavirus is a retrovirus. It works with RNA, and RNA, as it's being copied, you have all these errors that happened. And in many cases, you can see all these limbs, this tree, all these leaves of the tree. These leaves represent the variants of the virus. They're all slightly modified versions of the coronavirus. And the thing is, though, that they have become different just from mutations. And if those mutations helped, then the virus lived and it became a new variant. And then those variants became other variants, and other variants became other variants. And it just kept on going from that point. But one of the saddest parts about this whole thing here is that, remember, you've heard people talking about wearing masks. I've mentioned wearing masks. But the thing is, though, that if you can prevent the virus from moving around, then you can also prevent the virus from copying itself, because that means that no one else gets infected. If you're wearing a mask and you wash your hands, you socially distance, that means that you are preventing the virus from spreading. And every time the virus spreads, you have this chance of variants to be created. In other words, you have a chance of mutations to be created. And those mutations, if they help the virus, it becomes a variant. And if they don't, then the virus just dies, and then you got lucky. But in many cases, though, you can see that all of these limbs and these leaves that come off this tree are successful cases where the coronavirus was able to evolve in some way that somehow improved its chances of survival, or at least didn't hinder them. And so that's why we have these variants. And so by masking up and by getting your vaccine and by doing all these other things, you prevent these viruses from spreading or by becoming variants, because they can't copy. Anyway, this is just a lifestyle of the coronavirus. It's kind of an interesting thing, but it's the same idea as what we saw with influenza, where the cell has these, the virus has these protein spikes on the outside which stick to the cell, unlock the cell wall, the cell lets them in. Eventually, long story short, the RNA is then turned out of this virus. The RNA is then used to make protein. Protein then the protein helps to make some more DNA from that RNA, and then another long story short here, that DNA is injected into the cell. The cell starts producing more spikes and proteins for the virus, and then eventually the cell will just explode with all these pieces of these new viruses that will go out and start infecting other cells. In fact, if you look at where's that video? If you look at these videos here, check out this video here, the coronavirus life cycle. This life cycle will explain to you this chart in an amazing amount of detail. In fact, it's so much detail that it kind of makes you stop and think for a second, like, my gosh, this is a much more scary virus than I realized. Anyway, please do have a look at that thing, and let me know let me know what you think when we come together next time, or whenever it may be. Tell me what you think about that video. But remember, though, these are the spike proteins. This is another shot of the spike protein here, where the spike, I believe in this case, the spike protein is actually sticking to a human cell. The human cell has a receptor, which is the blue part, and the spike protein from the virus is the red part. I think my diagram is upside down, but I think that's how it works. And so when you have an antibody that sticks, for instance, to this blue or the red part of this spike protein on the virus, then that means that this spike protein cannot stick to the cell. It's blocked. It's like your spike protein is now kind of it's inoperable. It cannot operate on the cell. Um, Let me just move on to the next thing here. We have 5 minutes here, and I don't want to hurry people through here, but I really have to ask you to do this. This is not from me, this is from the college. I'm asking you to spend the 5 minutes that we have left in class to complete this midterm reflection, which must be done today. I mean, just at least click on this link now, and then you'll have it. But what I'm asking you to do, though, is to complete a survey, hopefully now or by the end of today anyway, but today. This is a midterm reflection, and if you look at this thing, really what it is it's just two questions which are asking you about your experiences in this course. I want you to be frank. I mean, the ideas that you put out are will help this class to become a better class.